And with that, we move right into our next segment, another exciting partner presentation. And we are pleased to have with us Cytiva, a global leader enabling breakthrough in life sciences and advancing the future of healthcare. May I now invite Mr. Mohit Chaudhary, Workflow Development Leader, Cell Therapy at Cytiva, for his presentation on Cytiva Cell Therapy Workflow. Welcome, sir.
Thank you for the warm welcome. I'm Mohit Chaudhary. I'm working as a workflow development leader with Cytiva. So I'm here to present the workflow and what Cytiva does and what is the role of Cytiva in manufacturing cell therapies and targeted therapies, which we are discussing since morning today. And it is quite an encouraging response from the medical fraternity, which has talked about to make those targeted therapies as a first line treatment. So it is again, as a manufacturer and service provider, it is very much encouraging for us also. So starting with this, and second thing, I will not take much of your time because the discussion would be brief in the interest of time. So first, what is cell? This is pioneering the future of the therapeutics. So the cell therapy has changed the way we are treating the cancers currently, especially hematological malignancies and solid tumors. So Cytiva has a key role to play in manufacturing those cell therapies because Cytiva ensures that these therapies are manufactured at a scale safely and with reproducibly. So that is the vision of Cytiva. Our vision is a world in which access to life-changing therapies transform human health, and our mission is to advance and accelerate therapeutics. So here is the journey of the cell therapy when it is developed from lab to the manufacturing scale, and Cytiva is able to support it at all the four stages from preclinical, phase 1, phase 2, phase 3, and commercial and post-launch. So here the reproducibility is very important because whenever a therapy fails, it does not fail because of the biology, because people understand biology very much who are in this field. But the actual reason of the failing the therapies is from the transfer from lab to the scale up. And this is where we play a key role, and we ensure that you get your reproducibility and homogeneous result at every step from preclinical to phase 3 and up to the commercial scale up and for future treatment. So as a size, we have 16,000 associates across 40 plus countries, 4 key hubs, more than 4,000 commercial associates serving 100 plus countries, 36 manufacturing sites, more than 100 plus flex factory platform and projects which we have delivered globally, 23 R&D and innovation centers across the world, and more than 96% customer satisfaction for services. So that is the size of Cytiva, which is spread across the globe. And India being a very important country for the Cytiva, we have set up a R&D as well as a manufacturing facility in Bangalore, which we call the Fast Track Lab, where we do validations and process also are developed by us there. So it is not only cell therapy when it comes to the advanced therapies. Cytiva is able to support to the viral vectors, nucleic acids, non-viral delivery, and mRNA also the part of Cytiva KT. So in most of the genomic, or we can say most of the advanced therapies are Cytiva has a contribution. So here is a cell therapy workflow, which is a typical CAR-T manufacturing workflow, which is provided by the Cytiva. So that is the workflow, and when it comes to the CAR-T manufacturing workflow, from apheresis to the final formulation and logistics, Cytiva has a role to play in every steps. So as a typical process, we get the sample post-apheresis in a frozen form or a fresh sample. Cytiva has an instrument called VIA-THA. With the help of the VIA-THA, it can thaw the sample. And then isolation is the first part where you try to isolate those T cells from the blood sample. So we have SUPEX instrument which can help you to isolate those T cells. And parallelly, we have other instrument also which we call Cephia Select. Then the next step is activation. So though the space is vacant here, but we have launched equipment we call as a Cephia expansion, which can fulfill that step also. The instrument is able to activate those T cells for the transduction. Next is the gene transfer. Again, the SAPEX CPRO system is capable to transfer the gene into the T cells and it make your CAR T complete. And parallelly Cephi expansion, which is not mentioned here, is again the second instrument which we have to do the same job. And when your CAR T is ready, you have to expand those cells so that you could give the enough amount to the patient. And we have bioreactor for that, which we call Zuri. It will do the job. It works with the help of some medias or cell growth medias, we can say. And then once you grow your media with the desired T cells with the desired volume, then it comes to harvesting. In the harvesting step, it removes all the impurities, which we can say, which has been added on the previous steps, and it will give you the pure T cells. And the final is the formulation where you can make your sample ready. And once your sample is ready, logistics part, there are a lot of modes of transportation with the LN2 containers, which will help you to transport those samples to the patient. And from there, you can inject to the patient. So if you talk about the entire process, Cytiva is capable of manufacturing the CAR T end to end. And more importantly, we have a software which is Chronicle. With the help of this software, all the equipment which we have shown in the previous slide can be synchronized with each other to monitor the process. Any problem happens, then you can get the alert also. You can store data and create batch records, mitigate risk by eliminating manual activities. So this is a very important thing when it comes to to comply with the regulatories. So this is what from our side. So just if any information you need, we have about that side. You can reach out to us if you are looking for some manufacturing thing in the CAR T space. You will be most welcome to us. So thank you.
Thank you so much, sir. Thank you so much for this presentation. I would like to invite Dr. Manisha Khurana. Ma'am, if you can please come on stage and felicitate Mr. Mohit Chaudhary for this amazing presentation. So, thank you, ma'am. Thank you, sir.